In this video, we will discuss how this Setd2 laws cause epigenetic changes in the tumors. So, what they will finally conclude is that this H3K27Ac deposition that is caused by Setd2 laws that leads to the epigenetic changes in the tumors. So, how they come up to this point, we will discuss these things through the results that are presented in this figure. So, from the literature, they identified that this H3K27Ac and H3K27Me and H3K36Me3, all these three are got dysregulated or are caused proliferations in the tumor cells. So they wanted to check whether these things are also get affected in the Setd2 laws derived tumor cells from KC from KSC tumors. So what they did is they checked the Setd2 wild type cells and did the Western blot on Setd2 wild type and Setd2 knockdown cells, and what they have find out is that there is upregulation of this H3K27Ac, H3K27Me3, and whereas there is downregulation of HK36Me3 in the Setd2 knockdown compared to the wild type. And these things they have shown also during the immunostaining, where we can see that there is an increase in this H3K27Ac signals and H3K27Me3 signals when the tumor cells derived from KSC compared to the KC TP3 tumors or KC wild type, and simultaneously they also see the decrease in this HK36Me3. So, and these things are quantified on the right hand side, where we can see that there is a decrease in the H3K36Me, whereas there is an increase in this H3K27Me and H3K27Ac. And simultaneously, along with that, they also find out that there is a upregulation of this acetyl CoA in the sorted populations from orthopathic tumors of the Setd2 knockout cells compared to the Setd2 wild type cells. And after that, they check this density of this H. So once they identified that acetyl CoA is upregulated in the Setd2 knockdown, that might help in the enhanced OXPHOS in the mitochondria, and with the help of fatty acid oxidation. So in order to do that, they further check this H3K27Me density and H3K27Ac density in the Setd2 knockdown and Setd2 wild type. And what they find out is that H3K27Ac is increased in the Setd2 knockdown, whereas there is not much change in the H3K27Me density. So this indicates that there is upregulations of this H3K27Ac in the Setd2 knockdown, whereas there is not much change in the H3K27Me3. And after validating that, after finding out this relationship, they check this RNA-seq data and find out the correlations between promoter gene body and intrigenic with the all genes. And what they have find out is that for this H3K27Ac gain, they see a relationship with all genes, whereas when they check it for this H3K27Me3, they do not see any significant relationship with all the genes. So it's this H3K27Ac gain has a role towards the all the genes that are upregulated. So right now they have established a direct relationship or proportional relationship between H3K27Ac gene and the upregulations of all the genes that are identified in those tumors. And now the did is they check the genes that are common in gene body and the promoter region, gene median promoter region, and also this intergenic regions, and the common genes that they have identified, and they wanted to check what are the using the geo analysis, what are the pathways or geo or CAG analysis of to find out the pathways that are related to this T39 overexpressed genes, and the pathways that they have find out is that ECM receptors interactions, glutathione metabolism, focal adhesions, and TGF-beta signaling. All these related to the proliferations markers, and also the oxidative phosphorylations is also increased. And after validating that these proliferations markers are increasing, they also did this RNA-seq and cut and tag analysis, and here also they find out the similar results, that is cell differentiation, oxidative phosphorylation, stem-related TFs, covalent endocytosis, and glutathione metabolism. All this related to the proliferations and migration of the tumor cells. And after that, what they find out that is the AP1 motif that is common in the genes that are upregulated by this H3K27Ac gain, H3K27Ac upregulations. And when they check this AP1 motifs in some of the upregulated genes, they find its similarity to this Gen B. Here we can see that when this H3K26Me3 upregulated, then there is a binding at the promoter regions occurs, but not at the gene body. However, for this H3K27Ac, when it's upregulated, there is not much binding in this promoter region, indicates that it will upregulate the translations of this GAV2 gene, whereas H3K26Me3 does not upregulates because it will interact with this promoter region and restrict the transcription of the promoter region. And similar results they have find out for the other genes that they have cross-checked out of the genes that are upregulated in this Setd2 cells. So this is how H3K27Ac helps in this upregulations of the genes, whereas H3K36Me3 downregulation of this H3K36Me3 also helps in the upregulation of the genes, as it will otherwise bind to the genes and affects the transcription of those genes. That is the reason the H3K36Me3 is downregulated, whereas other genes are upregulated, because their upregulation does not affect the promoter regions, and whereas in some cases it will enhance this gene body region. It will bind at gene body sites, and that is for very few genes, that is Kevin3. But for all the other regions, we can see that with the upregulations of this H3K27Ac, it does not bind to the promoter regions of the upregulated genes, most of the upregulated genes. So thereby it does not interrupts or hinders the transcription of those genes, and these genes are the markers for the proliferations, migration of the tumor cells. So after finding out these observations, they also did the in vivo study on the Setd2 wild type cells and Setd2 knockdown cells, and treatment is done every other days from the 7th day itself. And what they have find out in that case also, in the control case, they do not see a change in the tumor weight, whereas when they use JQ1, they see a decrease in the pancreas or body weight. That is quantified on the right hand side, where we can see that the Setd2 knockdown, they see a decrease in the tumor pancreas body weight. And for this lesion area also, they see a decrease in the JQ1 case. The JQ1 is used in the Setd2 knockdown cells, whereas there is no decrease occurs in the Setd2 wild type. And JQ1 is known to inhibit this acetyl CoA, and thereby inhibiting enhanced OXPHOS. So it will affect Setd2 knockdowns much, but there is not significant change in the Setd2 wild type. Indicates that acetyl CoA is the acetyl CoA has a relationship with this He3K27Ac formations. And they also check the other factors such as oxygen consumption rate, and they find out that oxygen consumption rates are lower when they use JQ1 in the Setd2 knockdown cells, and however when they use for the Setd2 wild type, they do not see too much changes. And compare the cancer-associated fibroblasts, which are rich in lipid, they are also downregulated when they use JQ1. That is, acetyl-CoA is dysregulated. This indicates that tumor cells epigenetic changes promotes further lipid tumor microenvironment. So dysregulation of this tumor epigenetic changes will prevent the lipid microenvironment formations. And they also check these relative expressions of the different genes of that indicates the markers for proliferation and migrations of the cells. And they are also decreased when they use JQ1. That is, CAPE2 is decreased, CAVIN3 is decreased, BMP2 is decreased, LDH1 is decreased, KLF5 is decreased. All these decreased when they use Setd2 knockdown cells and JQ1. Whereas in the wild type cells, they do not see too much, they do not see any significant decrease. This indicates that in the Setd2 wild type, the acetyl-CoA might be upregulated, that might causing the proliferation and migrations of the cells. And these things can be disturbed or dysregulated or inhibited or suppressed by affecting this by inhibiting this acetyl-CoA pathways using the inhibitor, that is JQ1. This is how they come to a point that the He3K27s got deposited because of the enhanced acetyl-CoA that will cause increased OXPHOS. And so this acetyl-CoA is deposited in the Setd2 knockdown cells, and that will results in the upregulations of the proliferations markers and migration markers. And simultaneously, this He3K27Me3 dysregulations also helps in the upregulations of these markers, proliferations and migration markers. So I hope you understood the relationship between He3K27Ac in the Setd2 knockdown cells that cause epigenetic changes. If you want to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.